So what did this new paper look at? The researchers took mesenchymal stem cells from psoriatic skin and compared them to healthy controls. Now, these stem cells are really interesting because they're not just passive bystanders. They actually contribute to the disease process and they respond to treatment, making them a potential therapeutic target. The team wanted to see what happened when these psoriatic stem cells were treated with Humira, a biologic that blocks TNF alpha, and they looked at the mitochondria in detail, looking at the morphology, reactive oxygen species production, calcium handling, and apoptosis markers, in other words, cell death. They looked at everything, and here's what they found. At baseline, psoriatic stem cells had very abnormal mitochondria. They were overly elongated, highly interconnected, pumping out excess reactive oxygen species and taking up too much calcium. And too much calcium within the mitochondria means they can't perform their usual functions of producing energy and cellular water. So basically, these were mitochondria under siege. When the cells were treated with Humira, something interesting happened. The mitochondria started to look and act more like healthy ones. Their shape normalized, their calcium handling improved. Even DRP1, the protein that controls mitochondrial fission, was restored to normal levels. Sounds good, right? Well, here's the twist. When the researchers added extra stress, either oxidative stress with hydrogen peroxide or inflammatory stress with LPS, Humira failed. It couldn't prevent mitochondrial dysfunction. The mitochondria slipped straight back into their abnormal patterns. And the drug also didn't rescue apoptosis signaling. The balance between cell death and survival remained off.